So is everybody kind of back? Okay. Okay, let's slowly get into it. So everybody got some nice brains on his computer? Some nice propagation, spreading? So actually I've done my PhD more on network science. And the propagation that you've seen spreading just by a single stimulus, spreading through the network. I had colleagues, most of my colleagues were working on epidemics, and it spreads very similar way, right? Just saying. So the propagation pattern here, it's propagating along a network. We just plotted it on the brain. But what you've just done could also be simulating an epidemic. Yeah. If you stimulate at the right point, you get a whole brain activity output. Pandemic. If you just have a focal point, you have contained your epidemic. Yeah. It's very similar. If you look at it from a network science perspective, it's the same. It's just a network, and it's a propagation of activity along the network. Yeah, so very, very current issues that we are dealing here with. And now let's get into entropy. So should I wait for people who's... Do you know anybody like right or left that wants to come back? No? Good. Okay, let's restart. Multiscale entropy. It sounds very fancy. It's not so fancy, actually. It's just an entropy at different time scales. What is entropy? Who has heard entropy before? Okay. Which one? I like this question. Yeah, which one? Because in general, we maybe all have heard of entropy, but we are like, okay, I'm confused. Is it water? Is it ice cubes in water entropy? No, it's signal processing entropy. I will get into that. It's quite an abstract measure, but what's the beauty about this measure is that it summarizes all the dynamics of your brain in one plot. That's nice, huh? I will show you these plots. So it's not on. Okay. Maybe this battery died in the meantime. That could be. We have another pointer. Huh? I like to have a pointer. Yes. Yes. Okay. Okay, now I see why Petra was doing this. Okay, no pointer.
Do you want me to put some batteries in them?
Yeah, okay. So as I said, the multiscale entropy is just a summarized measure of brain complexity of a person. So every individual we could assign a multiscale entropy to, and this is how complex your brain is. It increases in development from childhood to adulthood, some studies have found. So that should be a motivation for us to look at it. It's something important, apparently. When you're a child, your brain is very simple. The dynamics seem to be not that complex. It's a very deterministic system. And then you move in development towards adulthood. And that means you move towards a more complex system. And your brain is doing different things at different time scales, processing information all the time. So what's the long-term target here? Where do I want to get with this talk? I hope that in the end you will see that we could use this measure of multiscale entropy to predict if somebody will be suffering from a neurodegenerative disease or not. So in the end, we want to use it to predict the onset of dementia. Yeah, seems like a long way, but let's get into it. So overview of the next hour. So I will give about, I think it's half an hour again, kind of, a lecture about it. Please ask questions again. And then there's another Jupyter notebook prepared for you. And this time, not Mr. John Doe anymore, but real, not patient, but subject data here from Berlin, resting state data set. So that's pretty cool. First time we're loading in real data, not from like from a mouse or a monkey or something, from humans. Yeah, that's the con1-zip and con2-zip files. So I will first explain entropy, then introduce multiscale entropy. Then we will look at different studies who have used this, and I will show you some evidence that this is an important measure. It's something we should check out. And then I will show you it's not enough. We need two measures called local entropy and global entropy to actually characterize what's happening in healthy aging. because it seems not that easy, healthy aging. I will get to that. And then last is to calculate the multiscale entropy yourself, again in the Jupyter notebook, because this is then not available in the GUI yet. Yeah. Okay, so entropy, or the question why life always seems to get more complicated. You remember this, right? It's your room at home, it looks like this, and it's always moving towards this side. That's entropy. Always moving in that side, never the other direction. Yeah? It's just a natural phenomena, and it seems to be similar with our brain. It's moving towards more entropy. Okay, what does it actually quantify in terms of signals? Because what we are looking at, what we have been plotting, is these time series. So these time series you want to quantify, do they have a lot of entropy or not? How complex are they? And this entropy of signal processing entropy, if you have heard of other entropies, please forget them for now. Similar formula, totally different concept, okay? So here the entropy is quantifying the predictability of a signal, or rather the unpredictability. So entropy is high if a signal is very unpredictable. Entropy will be low if a signal is very predictable. So here we have an example of a very predictable signal. It's synthetic, just a sinus wave going up and down. Once you have seen this, you've seen it all, right? It's very predictable, comes again and again. So where do we put it on the entropy? 0. Not much information content there, not much entropy there, very ordered, just the same signal. That's it. That would be very low entropy. And then we have pure noise. That is supposed to be this one here. You have a lot of noise, and you say, okay, there's not much information, it's just noise. But if you just calculate the sample entropy, or the entropy of this one, it's actually placed quite high. Yeah, because there's a lot of stuff happening that's unpredictable. So if you just calculate entropy, it would be placed very high on the scale. And this is how maybe our brain waves will look like. Yeah, they will have some waves, but also some noise and so on. So they will be much more complex. And where they are placed in entropy is somewhere in between. So in between a completely predictable signal and complete noise. And when we want to then distinguish people who are all in this range, we need a different measure, not just entropy, because pure noise, as I just said, is scoring quite high on the entropy. That's kind of not good for us. We want a measure that's actually distinguishing brain signals with real information content from noise. So this measure is then called the multiscale entropy. And entropy is just calculated like this. It's a mathematical formula. You can remember it. It's quite nice. I like it, but it's because I studied mathematics. But if you don't like it, just think about it as a measure of complexity. Or high entropy means actually high unpredictability of a signal. Okay, so let's look at some more formulas. But if you don't like formulas, again, only look at this picture that explains it all. Here, entropy is always h. Actually, now I think, like, why is it h? I don't know. Maybe in another language, entropy is with h. But yeah, okay, it's always h for some reason. And that's the entropy of two signals, signal x and signal y. So everybody has a circle of its entropy. That's the entropy of signal x, entropy of signal y. And then if they're overlapping, here in the middle, this overlap is called mutual information. That's something mutual both signals have. So what does it actually mean? It means that there is a certain amount of predictability that I have of the signal x if I already know signal y. That's a conditional entropy here. So just think about it. You want to predict signal x. Yeah, you know signal x has a certain amount of unpredictability. Then think about, you already know what y will be doing. So that takes this uncertainty away from signal x. And all that's left is the uncertainty of signal x after knowing already signal y. This is how it works somehow. So when you're looking then at the mutual information, it's nothing else than the whole uncertainty of signal x, the whole circle, minus actually the uncertainty that is taken away from it when we already know why. Whole sphere minus this one, what's left over is the overlap, the mutual information. Mutual information means the signals are somehow in relation with each other. So when two signals are independent, how would the circles look? Yeah, I like that many people are doing this. Yeah, exactly, no overlap, no mutual information between two independent signals. So how is it in the brain? Nothing's independent. All signals are related to each other somehow. There's always some overlap. When you know one, you can predict the others, but not completely. So yeah, that's why entropy is maybe a good measure to take a look at. Okay, so the joint entropy, okay, that's another formula that you can just see from this. If you want to have the joint entropy, so the uncertainty of signal x and y together, you want to have the whole space together, you just take the conditional ones plus the mutual information. That gives you the two beautiful circles together. Okay, that's just a visualization of the formulas. Actually, we will later on have a Python function for you ready. So then you don't have to remember this. But that's where it's based on, the whole concept. And I mentioned already that this can be important for the brain, and why. We will look now at entropy at different time scales. So why are we doing this? Because the brain, this matrix here is the standard connectivity matrix. We know it all. We have seen those pictures, hopefully today, yesterday, we plotted it. And this is actually all the one-to-one connections. So everybody I can reach on a direct way. But then there are different other matrices here. And that's actually that because the brain has on different spatial scales, it has different paths that I can go and reach. So I not only have direct connections, but I have indirect connections, friends of friends, neighbors, and so on. So on different time scales, I'm able to reach different other areas. I'm able to influence different other signals if I'm a neuron. So the brain is much more complex, right? It's not just always this time scale and then it kind of dies and again pops up, dies and again pops up. No, it's multiple processes happening at different time scales. At the moment you are watching, maybe also memory processing, I hope still, and at the same time auditory. Multiple processes happening at different time scales. So we would expect for the brain to have a very specific behavior towards different time scales. The entropy on different time scales should look different, should not be all the same. Maybe we have something specific there. So this is how they got to this scale of multiscale entropy, which is then just calculating the entropy for every possible different time scale. So how do they do it? With coarse graining. So we first have a time series. We have every second one signal. 1, 2, 3, 4, 5. This above signal of the axis, we can just calculate the entropy of that. Then we know this is the entropy of brain region X, full stop. But now we look at different time scales, and we do it like this. This is called coarse graining. We just average over always 2 points, x1 and x2. We have a new variable, y1, y2, y3, y4, and this gives us a new signal. And we calculate the entropy of this signal. It's the entropy of time scale 2. And you see where I'm getting at, huh? Don't have to do it again, probably. This is then averaging over 3 points always, and this would be the entropy of time scale 3. So we look at different time scales. What's the brain doing on different time scales? And then let's not yet look at the brain, but let's look at noise. Because that was the reason why we introduced different time scales. Because we said pure noise has also a high entropy. So we need a measure where we can clearly classify noise from our brain. That we can say, okay, this is very high, this is good for us, but it's not pure noise. Huh? We don't want to have something that distinguishes between pure noise and the brain. And that's exactly this multiscale entropy. So here's the first plot you see of multiscale entropy. Many will come. It's always the entropy measure and the scaling factor. Time scale 1, 2, 3, I've shown you before. And here's white noise, for example. So the white noise here has a very specific behavior over the different time scales. We remember that it had quite a high entropy when we were looking at time scale 1. But you see, the further away I step from the time series of noise, I look only at the 2 points, I look only at the 3 points, 4 points, oh, I look only at every 15th point, I see that the entropy goes down. So the unpredictability of that signal goes down, because the further I step away, maybe you can think of it like this, the more I see is just noise. And white noise has this specific behavior. There's another noise, that's 1 over S noise. It's actually an inverse noise, inverse to the frequency. You don't have to worry too much about it, but let's say it's a much more complex version of noise. So you wanted to have more information content. So that's why it's nice that it has also higher entropy. And you can clearly distinguish this noise from this noise, because here you see over the different time scales, this more complex noise stays constant. So that people have looked into this theoretically and have said, hey, I've looked at this noise, and Julie in the back has looked at colored noise and multiscale entropy, for example. You can do a lot of theoretical stuff on it, but we want to look at the brain, of course. That's why we are here today. So we hope that truly complex signals have some timescale dependencies, that have some nice behavior, that will be also distinguishable between persons. That would be ultimate goal. And showing high entropy across different time scales. Here's one more example for you. A time series looking like this. You say, okay, it looks like a sinus curve, not really. It has maybe some more variation in there, right? So it has not an entropy of completely 0, but it has quite a low entropy. And over different time scales, we see it's kind of constant. And this in comparison here, which looks now a bit more like a brain signal, has high entropy. Yeah? That means it's not completely predictable. It has something in there. And also over different time scales, it seems to be consistent. That's just also a synthetic signal here. That's just for explaining. Now let's get to a hypothesis for the brain. So babies, grandmas, and you somewhere, I put you on the highest point, in the middle. So let's hope we are on our most complex point of our whole lifespan. Because in development, they have shown, I will show you in a minute, that your complexity of your brain seems to increase. And then naturally, what you would think of is maybe when you're getting older, healthy aging, your complexity decreases. Could be a simple conclusion, right? It's much more complex, but this would look the nicest way, I think, yeah. So multiscale entropy should somehow increase and then decrease. But anyways, in all this lifespan, you would hope that there would be a scale dependency. It's not always constant, but at different time scales, your brain is doing different processes with different entropy. That's what we are hoping for. So the first real brain curve here with multiscale entropy. Different time scales, you've seen this before, and the entropy always calculated. And what have they done here? They have done an experiment, and they measured EEG during a task. What kind of task? Viewed individual faces in one back memory. They always showed you a face, and they said, does the current face match the one you just saw? That's kind of like Tinder, right? And you're like, huh, that's an error. I've seen that person before. That's what you do all the time. Maybe, I don't know. So that's a very simple task, but they showed it to children. And children and adults. So the adults, let's make start with the children. What happens? A lot happens, quite a good, like a high evoked response, and then nothing else beyond that. If you're a bit older, you see there's some secondary process going on here, on a different time scale maybe, and the amplitude decreases, but you see that some other process is also visible here. And for adults, the amplitude is very low. What happens? But you see that there are probably like 3 processes going on. So you're doing something more than just this, no, that's not what I've seen before. But something else is happening. Maybe you're also remembering more or processing it more. Who knows? But this is the response on EEG. And then when we look at the entropy here, we can clearly see that the adults, and this nicely goes up. The older you are, the higher your entropy, the more complex your brain signal, and also over different time scales. Yeah. So that's what we wanted to see. Yeah. We wanted to see that the entropy, your brain gets more complex. So that's what they found with this experiment. You think, okay, now we've known it all, and then it goes down, right, when you're getting older. No. Then what they found was this. You have young people, middle and older people, and you see up till here, it's what we saw. The older you are, the more complex your signal is. But when you're aging, it seems they're overlapping and flipped. At a certain time scale, you seem to be then less complex than younger people. So what's happening here? And actually, they had the hypothesis, and what they showed is that two different processes are happening in healthy aging. It's not only that your complexity goes down, but your brain is doing something, I think, very, very smart. Because it's getting more effective. What do I mean with this? So it seems that your brain is processing memory and stimuli more locally when you're older. Not using the whole brain anymore, but processing locally more and less globally. And that makes it a bit more effective because you're not using the whole energy to process everything globally and compare this with, I don't know, everything in your brain, but you're processing very locally. And then you're like, okay, I just have to do this one back memory task, so I'm just thinking about the last 5 seconds or whatever, and again, and again. Yeah, maybe like a machine or something, I don't know. So that's what they found, that there is more local dynamics going on and less global interactions. And this process even seems to be coupled with better cognitive performance. So if you're actually better performing on tests, if you are processing information more locally and less globally, and a more active lifestyle. So the more active you seem to be when you're aging as well, the more your brain is actually processing only locally and not always using the whole brain doing some global processing. And then even what is worse now is that this shift, when you're going more and more complex dynamics, and then somehow your local dynamics, they change. This kind of shifting towards local processing instead of global, seems to be very important. And if you don't have that shift at some point in your lifetime, this seems to be predictive for developing dementia. I will say that again. If you're not shifting to local dynamics at some point in your life, this seems to be predictive of developing some neurodegenerative disease. So it seems to be important. So I will show you the empirical results they have for this. How did they calculate this local processing and global processing? I already said we calculate multiscale entropy. It doesn't seem to be enough. Curves are overlapping. So they developed two new measures. First of all, the local entropy. That's just for every region, I. You look at all other regions, and you look how much uncertainty are they taking away from me. If they are taking away all uncertainty here, and this would become completely certain, yeah, then there's not much local entropy going on. But it seems to be locally quite complex. Locally, there's a lot going on. It's increasing in aging, local entropy. And increasing was blue ones, yeah. Here underneath are all the regions they listed. This is EEG again from the middle, older, and younger group. And everything that's blue here, and that's basically everything, is all the regions that increased in local entropy. So locally, your process becomes more complex. But globally, or how they call it, the distributed entropy, it's going down. And the distributed is the mutual information between two areas. So how can you think of this? Oh, sorry, I bumped against this. Okay. How can you think of this distributed? That's like this area versus area. And then if they have some mutual information globally, it's blue and it's increased. And you see that within a hemisphere, this is within hemisphere, and this as well. You see that we have in aging, we have an increase. So meaning that within a hemisphere, regions are working more together. There's more mutual information, more overlap. But cross-hemispheric, those two, it's red. Red meaning actually a decrease, which is happening much more here, meaning that cross-hemispheric means going from one to the other hemisphere. If there's not much mutual information, there are different processes going on, not the same one in both hemispheres. So this is what they mean with going more to local dynamics. Going more towards me and my neighbors, and in my hemisphere I'm processing information, not globally. So they say aging shows a strong cross-hemispheric decreases in distributed, so in global entropy, and some increase within a hemisphere. So you somehow get better when you're aging at local processing and local dynamics. You're getting less good at global dynamics. So different processes happening. So then they said, okay, it's not only aging, but we have to distinguish within our older adults, we have to distinguish high-performing adults and low-performing adults cognitively. That's where we are getting at. Because it seems to be related with cognition as well. Because when they split, the high-performing and the low-performing older adults men, so same curves here, this gray one is split into blue and black. And when they had the blue and black curves, they saw, oh, it's actually different if you're cognitively still fit and if you're cognitively not so fit anymore. You see that's different? Blue and black. What is different? We see on finer time scales a peak, and on coarser time scales we see less good performance than young men. So we see this shape for high cognitively fit people. And here we see it again. If we split the group in the ones who failed the test and the ones who passed the test, the ones who passed had a peak, and then at larger time scales, a decrease. So it seems to be a very specific profile that a cognitively well-performing person has when it's aging, when he or she is aging. So now this is all healthy people. Now you want to know, okay, so that's cognition and some cognitive tests for elderly people. Nice. I want to know if I get dementia or not. So they looked at patients. And look at these curves and just have in your head maybe a picture of how patients should then look like if they're somehow abnormal. Okay. And here it is. Here we have controls, green line. That's normal here. We assume they're healthy, having a normal shape. And then we put on, and we have different groups here. They looked at Parkinson patients. There are two different groups of Parkinson patients. One are developing dementia, PDD, the red ones. And ND is non-dementia. They're non-converting, not converting to dementia. So what's the difference here? Can somebody tell me? Red and yellow curve. What's different? I know it's late, but we can do this.
Yellow one doesn't peak before it starts to drop.
Yeah. A little bit, huh? Yellow one peaks here a lot, and then these are actually two different areas in the brain. Yeah. So we are actually interested in the ones that are converting. So the red one are the ones, the Parkinson disease patients that are developing dementia. And this is before they develop dementia. So they're not peaking at the finer time scales. They're below. And then they're not decreasing at the coarser time scales, just as you said. They're above, below, above. Not a very healthy cognitive profile, looking at their multiscale entropies. So they're looking like the people who performed not so well on cognitive tasks. But this is not task data. This is just resting state.
This is
just resting state, no task. And you already see this, that they're clearly having a different profile of multiscale entropy. It's the first study. It's the first study, and they looked at some Alzheimer as well. I don't show that, but it seems that they didn't have this shift of local global entropy. And now the most recent study, published last week by our collaborators. So what did they do here? No more diseases. They had young, healthy men from 18 till 35, just like we're here. And then they played them some music, beautiful music, different songs. I think it's more like opera-like. And then, yeah, I don't know all of them, but quite a few. Okay, and then they gave them a task. So they gave them two tasks, emotional task and perceptual task. The emotional task was, how does the music make you feel? Think about it when you're hearing it, yeah? Just imagine, you're a young man, you're giving this task. They're giving you this task. And then they're measuring your brain activity. And the other was perceptual songs. So they gave them these songs and they said, is this instrument in there or is that instrument in there? So something you also had to think like logically about. So can you see a difference here between the green curves and the red curves? I think green and red is not very smart if you're like red, green, blind, right? It should be all gray to you. And that's what young men often have. Okay, so I will help. Let's look at this curve here. It has a high peak, and that's actually a perceptual curve. So if you are performing the cognitive task, this is the curve you wanted to have. You have a high peak and you have a decrease. That's a good one. Yeah? High complexity, something really high is happening. And then the emotional songs, something is happening like at a larger time scale, but here they are going higher than the green ones a bit. So there seems to be some other process also going on when you tell people, think about how this song makes you feel. Yeah? So different processes going on when you're looking at it emotionally. And how they concluded in the paper is that emotional tasks have some additional internal information processes apparently going on. Not just high peak then going down, but something else is going on. And that's something beyond the external stimulus going on in healthy men brains. In other words, a brain atlas is something, the brain adds something when judging the emotional value of music. That's a beautiful conclusion, I think, for something that we see here, which is pure mathematics, right? Okay, so maybe you see now why a future project, for example, of ours is if we can use the multiscale entropy to predict the onset of neurodegenerative diseases in large cohorts. And of course, we want to use TVB, right? Because we have seen that different music that is playing, different tasks that you are given, will trigger some different response, a different curve. So we can't give people all the possible tasks. So why not do it on the visual brain, giving different stimuli, like an auditory, a visual stimulus, and so on, just checking out your brain there. Then you don't have to sit under an EEG cap for too long, right? That would be the ideal. And then looking how complex is your brain, and then judging. Either you had this shift at some point in your life, or you didn't. And if you don't have it, you should work for it. Yeah. Okay, that was it already. Are there questions for the lecture part? Yeah.
So these plots that you showed are all averages across participants, right?
Yeah.
Grouped by either condition or type of participant. But like, how wide are the confidence intervals?
They're pretty wide.
So the sensitivity and specificity is actually pretty low?
So far, the studies had not such big cohorts as I've seen. So it's quite interesting. Actually, Paul has done lots of simulations on the cohort here, which is then a bit larger, and then still quite large confidence intervals. But maybe that also depends if you're looking at resting state activity, because then you're not really sure what kind of processes are going on, instead of giving people a specific task and then looking at their information processing. So it seems to depend highly on what kind of task are you giving, or whether you're leaving them in resting state. And when you have resting state, what I can judge just from the error bars looking, it seems that there are larger error bars in resting state, because so many, for different people, different information processing processes are going on.
Yeah.
That makes a bit of sense, I guess.
But do you have like a sense of how strong of a measure it is in the clinical context?
No, not yet. 2 small cohorts, I would say. But I would have to look that up as well. And since you're my colleague, we can easily do that. Yeah, and it's current project, right?
Okay.
Yeah. Any more questions? Yeah.
So you mentioned cognitive tasks.
Yeah.
What cognitive tasks are you? It's a broad thing, right?
Yeah, definitely.
What do they actually check?
The MOCA test. So I would have to look that up. I'm not a psychologist,
And
no.
also
But the references are all here, so you can check out. It's the first is a good summary of everything. It just came out on the. I think it's now
also
published.
the music pieces, some of them are very famous. It is possible that they recall, but I don't think anyone knows Glassworks or stuff. So it is very, yeah. No, I mean not everyone, right?
Not to insult anybody.
No, it's amazing piece of music. But yeah, this is my problem. It's very interesting, but it also seems like very early.
It's very early.
Storytelling,
Yeah.
kind of.
Definitely. So there's hope, I would say, and not yet so many results. But that's why it's a cool area to get into, because whatever you find can be leading in the field, right? That's how I see it, as an opportunity as well. And never used TVB for these experiments before, so let's check it out, I would say. Okay, more questions for now? I would just then shortly otherwise explain the tutorial again. That's the last thing we are going to do today, right? We are all the way here, below here, tutorial here. And there's a notebook prepared for you as well, but I also made a stepwise thing here. If you are now very brave and you say, I can load this data by myself, it's just region-based, no surface involved anymore. You can do surface, but we don't have the individual surfaces for you yet. So you can load the data from the 2 persons. They have different ages. And then you decide what kind of stimulus you want to give. Feel free to do something crazy. I put something in the Jupyter notebook, but you can do whatever you like. I think I put something similar than the last stimulus. You can also do resting state, whatever you like. And you can calculate with the function sampn, that is actually, you can just load from the TVB box. You can then make those beautiful multiscale entropy curves. And then hopefully get different curves, I hope so, for the 2 different persons, because they have different ages. We have a 30-year-old person and a 68-year-old person. But it's early stages, so let's see. And you can also look, I put some additional tasks as well. You can look what our default John Doe, how complex is his brain, right? You can just load this as well, look at it. And also different stimuli, maybe they get different curves. So I tried out, they give different curves. So that could be also nice to look at. So again, the choice is yours. You do. So this cannot be done in the GUI, the multiscale entropy. If you have been working on the GUI, this is the moment where you should completely switch to the Jupyter. Look at the notebook. Try to load the data, and I think that's the most exciting part, because when you can do this, you can load your own data, and that's what maybe some people are going for. And then you can check out your own data and get those curves. Okay, and we'll do it again the same way. How late is it? It's 4:15, so if you're fast and you understand everything perfectly, you're of course just allowed to leave. Right? And if you feel like, oh, I rather would do that at home, that's also completely fine, right? You can ask questions tomorrow as well, but we will be here until the last person leaves, or at least me. Okay, good luck.